Good morning, everyone, and thank you for being here. Today, I'm excited to share with you a fascinating topic at the intersection of nanotechnology, polymer science, and biomedical engineering: pectin-based nanoparticles, their structural features, synthetic strategies, and growing applications in the biomedical field. This PPT systematically sorted out the basic overview and chemical structure of pectin, elaborated on its various preparation methods and synthesis optimization strategies, explored its material properties in combination with a variety of characterization techniques, and focused on the application potential of pectin in drug delivery systems and its wide range of uses in other biomedical fields. At the same time, it analyzed the advantages and challenges it faces in practical applications, providing a comprehensive reference for the in-depth understanding and development of pectin-based functional materials. Due to their excellent safety profiles, biodegradability, and functional tunability. Among them, pectin stands out as a promising candidate for developing nanoscale drug carriers. It's a polysaccharide primarily extracted from fruit waste, citrus peels, and apple pomace, and is well known in the food industry for its gel-forming ability. But beyond that, its structure offers rich functional possibilities that make it highly adaptable for biomedical use. Chemically, pectin is composed of alpha-14 linked digalacturonic acid units with functional groups such as carboxyl, COH, and methoxy, OCH3, along its backbone. These groups are central to pectin's gelation behavior, particularly in the presence of divalent cations like calcium. Depending on its degree of methylation, pectin is categorized into high methoxyl and low methoxyl types. The low methoxyl variety, in particular, enables ionic cross-linking under mild conditions, facilitating the formation of hydrogels and nanoparticles without harsh chemicals or extreme pH. When it comes to synthesizing pectin nanoparticles, there are multiple established techniques. One of the most common is ionotropic gelation, where calcium ions are used to ionically cross-link pectin molecules, resulting in stable nano-sized structures. Another approach is nanoprecipitation, where pectin is dissolved in a solvent and rapidly added to a non-solvent to trigger the formation of nanoparticles. Emulsion solvent evaporation is yet another method, where pectin is first emulsified in an organic phase, and as the solvent evaporates, particles are formed. Additionally, ultrasonication can be employed to reduce particle size and improve uniformity by breaking down larger aggregates through acoustic cavitation. Each method has its own advantages and limitations, and the choice depends on several factors, including the nature of the drug being encapsulated, desired release kinetics, and production scalability. However, regardless of the method, a crucial step in the development process is the optimization of synthesis parameters. Factors such as pH, polymer concentration, ionic strength, stirring rate, and temperature significantly influence nanoparticle characteristics such as size, surface charge, encapsulation efficiency, and stability. For example, maintaining the pH in the range of 4.5 to 5.5 often results in more stable gel networks and better control over nanoparticle morphology. After synthesis, the nanoparticles are subjected to rigorous characterization. Techniques like dynamic light scattering, DLS, provide insights into particle size distribution and polydispersity. Transmission electron microscopy, TEM, allows us to visualize the particle morphology at the nanoscale. Zeta potential analysis is used to determine surface charge, which directly affects the stability of colloidal systems. In addition, Fourier transform infrared spectroscopy, FTIR, can be applied to verify the presence of functional groups and confirm the interaction between the polymer matrix and the drug molecules. For evaluating encapsulation efficiency and release behavior, UV-vis spectroscopy, or HPLC, is typically employed. So, what makes pectin nanoparticles so relevant in drug delivery? First, their biodegradable and biocompatible nature makes them safe for systemic administration. Second, they can be engineered to be pH-sensitive, allowing for targeted delivery in regions such as the intestine, which is especially advantageous for oral formulations. Third, they can encapsulate a wide range of therapeutic agents, from chemotherapeutic drugs like 5-fluorouracil to biologics like insulin, as well as nucleic acids, antibiotics, and antioxidants. Their ability to deliver these agents in a sustained and localized manner helps in reducing side effects and improving therapeutic outcomes. Beyond drug delivery, pectin nanoparticles also show strong potential in other biomedical domains. In tissue engineering, pectin-based hydrogels serve as excellent scaffolds, supporting cell adhesion, proliferation, and differentiation. Their tunable mechanical and biochemical properties allow customization for different tissue types. In the field of antimicrobial therapies, pectin nanoparticles can be combined with silver ions or antibiotics to produce coatings that inhibit bacterial colonization on medical devices. Furthermore, pectin is being explored in functional food systems, where nanoparticles enhance the bioavailability of poorly soluble nutrients or probiotics. In cosmetics, they are used for the controlled release of active ingredients, providing prolonged efficacy while minimizing skin irritation. Of course, while the promise of pectin nanoparticles is substantial, there are several challenges that need to be addressed before widespread clinical adoption. One of the key issues is batch-to-batch variability, which arises due to the natural origin of pectin and the influence of source material and extraction method. This inconsistency can affect physicochemical properties and thus reproducibility in formulations. Another challenge is the stability of pectin nanoparticles during storage and after administration. Since they are highly hydrophilic and sensitive to environmental conditions, maintaining their integrity over time can be problematic. In addition, scaling up the production process from lab to industry poses both technical and economic hurdles. Regulatory approval may also be complicated by the lack of standardized guidelines for complex natural nanocarriers. Nevertheless, research is ongoing to overcome these limitations. One promising direction involves developing composite nanoparticles, where pectin is combined with synthetic polymers or other natural materials to improve structural integrity and functionality. Another area of growth is the design of stimuli-responsive systems, where pectin nanoparticles are engineered to respond to environmental triggers such as pH, temperature, or enzymes to achieve more precise and intelligent drug release. In conclusion, pectin nanoparticles represent a compelling platform for next-generation biomedical applications. They offer an effective blend of safety, sustainability, and functionality. As we continue to improve formulation strategies, optimize synthesis, and expand our understanding of their in vivo behavior, we move closer to translating these systems from bench to bedside. Thank you for your attention. I'd be happy to take any questions or hear your thoughts on potential collaborations or ideas to explore further applications of pectin-based nanocarriers.